Cancer is a result of genetic mistakes, genetic aberrations.
Cancer is unbridled growth of normal cells that is not acted upon by normal growth control mechanisms.
Cancer is probably the outcome of the processes that led to us being on Earth. If there was no such thing as mutation, if there was no such thing as genetic selection, if there was no apparatus that could promote cells to behave in the cancerous way, we'd still be single-cell organisms swimming in a sea.
Cancer is our normal life processes gone crazy. In the early 1970s, when I began my career in oncology, our strategy was what I call seek and destroy. Find it and kill it. And find it as early as you can and kill it as dead as you can.
Heretofore, we used chemotherapy, which was kind of like a sledgehammer. We'd kill the good guys and the bad guys together and hope that the good guys could recover.
When I was an intern, I took care of a woman who was 42 years old, just the nicest person in the world. Her leukemia eventually relapsed. And we tried more chemotherapy, and it induced basically a complete eradication of her bone marrow. We got rid of the leukemia, but we probably got rid of most of her normal blood cells as well. And she developed an infection as a consequence of that, as many people do. And she had these very high spiking fevers. She was very sick one night, and I called her husband and said, I think you ought to come down and bring the kids with you. And he came in to see her, and I said, I'm not sure how much longer she's going to be able to go like this. She saw her kids and had a chance to say goodbye to them. And then a couple hours later, she died. And you do the paperwork and talk with the family and do the best that you can to try to make sure everybody's okay. And then I just went to the call room and I just had a really tough night. And I just swore there had to be a better way to do this.
In the closing decades of the 20th century, a scientific revolution transformed our understanding of basic life processes. The new field of molecular biology shed light on the interior world of cells and the genes within them. We now know that these genes, small particles of DNA, control an ongoing production line of chemicals that determine the structure, growth, maintenance, and ultimately the death of all living organisms. This knowledge has altered our view of cancer, leading to new forms of cancer treatment. In the third program of this series on cancer, we look at some of these treatments, how they were discovered, how they work, and the promise they may hold for the future.
I disagree with the term a war on cancer. That's a baloney term. It's a public relations term. Basically, what you're doing over time is bit by bit, people are learning about cancer. And bit by bit, we are using that knowledge to better treat leukemia and other cancers.
Well, I think for sure in the history of mankind, we're at the most exciting juncture in the development of novel strategies for approaching the cancer problem, whether they're prevention strategies or improved diagnostics or enhanced treatments. There's just no doubt that the remarkable fund of basic science information that's been accumulated in the last several decades and is continuing to emerge forms a remarkably exciting, fertile ground on which to think about improvements for the care of patients.
The big hope for the future is that if we understand the molecular defects inside cancer cells, we can target them, and we can create drugs that specifically interfere with those defects, allowing us to kill cancer cells selectively while leaving normal cells untouched, thereby reducing undesirable side effects. And there have been several home runs that have been scored in recent years using this rational drug design. Gleevec is perhaps the best known of these.
In May 2001, the Federal Drug Administration announced the approval of a new drug designed to treat a form of cancer called CML, chronic myelogenous leukemia. The drug was called Gleevec. When Gleevec's approval was announced, Albert Willis had already been taking it for almost a year as part of a preliminary clinical trial.
I remember just being a Friday night, I was watching the news, and there was talk about this new drug that had been in the phase 1 clinical trial, and how effective it was in this small group of patients, and how it was very promising for people with my type of leukemia.
There was a buzz in the internet, in the CML community, that was like none other I've ever seen. And because of the rapid way we have to disseminate information, patients often led their doctors to Gleevec. They went to their local doctor. I have CML, I heard about Gleevec, where can I get it?
Albert's doctor referred him to the Brigham and Women's Hospital near his home in Boston, where a clinical trial of Gleevec was underway. Albert volunteered to participate.
It was sort of like a trade. I understood that I was going to be participating in tests, in some experimental drug, in return for, being able to take this drug and seeing if it's going to have some positive results, either for me or for other people.
Right.
So
It's interesting, though, too.
The Boston study was directed by two physicians, Richard Stone, a clinician who works mainly with patients, and Gary Gilliland, a scientific investigator who works mainly in the laboratory. This combination of science and patient care is called translational research.
Translational research is a word that many of us academic doctors use to talk about something that comes from the laboratory to the patient. Another word for it is bench to bedside. Bench referring to the laboratory bench where the experiments are done, and bedside referring obviously to patients.
The Gleevec story is just a wonderful story of laboratory bench to bedside research. It was largely spearheaded by Brian Drucker, who's a good friend, colleague, and he took advantage of the insight that CML was caused by a mutation in one gene. Out of the 35,000 genes that we have in each cell, one gene is defective. And he identified a compound that was able to take that gene and basically convert it back into normal function. That's what Gleevec does for people that have CML.
To really put the first few patients on Gleevec and to see what can happen was really very personally moving and dramatic, because we had patients who had CML who were not responding to the available treatments. In some cases they were very sick and we didn't think they had very long to live. And we placed them on this pill, Gleevec, and in many cases, in a matter of days, their blood counts improved. They improved in the way they felt, and in some cases this continues to this day.
The scientific understanding of the cause of CML began in the 1960s, when two doctors in Philadelphia noticed that the cells of all their CML patients had a common defect. One chromosome was missing a short segment of DNA. It came to be called the Philadelphia chromosome. 10 years later, using new techniques, another scientist found that this short segment of DNA came about because pieces of two different chromosomes had traded places. This is called a chromosomal translocation. This altered DNA in the Philadelphia chromosome affects a protein called ABL, which tells a certain type of white blood cell to reproduce. This alteration causes the ABL to lose its off switch, so the cells reproduce endlessly, eventually causing death. This uncontrolled reproduction of white blood cells in patients with the Philadelphia chromosome is chronic myeloid leukemia. Scientists discovered a small region in the ABL protein required for its uncontrolled activity, and this small region became the target for treatment.
The drug Gleevec has been designed by a drug company to specifically insert itself into the ABL enzyme and to interfere with its functioning, thereby depriving these leukemia cells of the growth stimulatory signals that this oncogene protein has been emitting. And that in turn causes these leukemic cells eventually to stop growing and even to die.
It took another 13 years of laboratory and animal research before Gleevec, then known as STI-571, was ready to be tested in humans. Research scientists at the pharmaceutical company Novartis drove the development process in collaboration with outside scientists.
One needs the resources of a large drug company in most cases to develop a drug in oncology today, because they have the resources to develop the drug from an idea to preclinical testing in cell lines and animals to make sure it's reasonably safe in animal models, so that it's reasonable to test in a patient.
Pharmaceutical companies are very enticed by the notion that they can help people with these diseases and that it can be a market of reasonable size for them, so that it's worth the time and effort to develop the drug.
I tolerated it very well. I didn't have any, side effects. Nothing very serious. A little nausea here and there. But
The tests of Gleevec in humans began the traditional way, with a phase 1 study on just a few patients to learn whether the drug was safe. It was. The next step was a series of phase 2 studies involving many more patients, including Albert Willis, to see if the drug was not only safe, but produced some clear benefit, such as shrinking the tumor or delaying its progress.
And this is really the watershed moment in the process, because if a drug enters its phase 2 program and then shows good evidence of activity, then that becomes a very clear basis to move on to the next phase, the so-called phase 3.
The phase 3 trial is the one that's really required to prove that it's going to be approved, because a phase 3 trial compares a new drug to an old drug in a randomized way. So half the people get new drug, half the people get the old drug, and we see which is better.
If the new drug produces better results, FDA approval usually follows, and the experimental treatment then becomes the new standard. But with Gleevec, something different happened. The FDA applied a new fast-track procedure and approved the drug even before the phase 3 results were in.
With Gleevec, it just seems so darn easy to figure out that it was going to be good. Do we really need to go through all that rigmarole to get it approved? And so basically the FDA said the phase 2 trial looks good. A lot of people are getting, their counts are getting under control. Many people's Philadelphia chromosomes are going away. We think we'll approve it. I think it's a wonderful resource.
Yeah.
We are working very hard to try to shorten the timelines, for example, from when a trial opens to the point when you have had sufficient participation and accrual to be able to provide an answer.
you need to go to you need to expand your stuff, like go to.
Albert Willis has been taking Gleevec for almost 5 years now. He's done well on the drug. His symptoms are gone and the side effects are minimal, but half his white blood cells still carry the Philadelphia chromosome. The so-called miracle drug has kept his illness in check, but has not succeeded in wiping it out.
I've had what they would describe as a partial response. So basically, my blood counts are normal now. In fact, as my doctors tell me, just taking a blood test, nobody would know that I have leukemia. What is still happening is I have a Philadelphia chromosome marker in some of the cells. So basically, I'm in partial remission.
The drug's only been around since 1999, really. So we still don't really know if it's curing people. If a person has been taking Gleevec for 3 or 4 years and they seem to be still doing very well, if you can't detect the CML cells, that's great. We don't know how long that's going to last. And then there are some patients in whom clearly it's not working that well because their blood counts are no longer controlled in the way we want them to.
Albert's response to Gleevec was only partial from the start, but other CML patients went into complete remission on Gleevec at first, only to become resistant to the drug later on. It's now known that most patients who already have advanced disease when they start taking Gleevec will relapse within a year.
As for any antibiotic, which is what Gleevec essentially is, we could have predicted, and now it's been observed, that these leukemias can figure out a way to get around Gleevec. They can develop resistance. So the challenge is to try to figure out what to do then or how to prevent the development of resistance.
Scientists are trying to overcome that resistance by combining Gleevec with other drugs, some that already exist and some that are still in development.
In cancer, we've always taken one drug that's worked and combined it with another drug that also works, and that's what we're doing there. There are chemists at these drug companies who are synthesizing new drugs which may be able to kill a Gleevec-resistant cell. And just like the drug companies which make antibiotics have come up with antibiotics to work where a given one doesn't work anymore.
Drug resistance is just one aspect of the research explosion that Gleevec has inspired. Scientists worldwide are seeking to create new Gleevecs active against a whole range of cancers, including acute myelogenous leukemia, a rare stomach tumor called GIST, and glioblastoma, a type of brain cancer. There is also hope that Gleevec-type drugs will work with solid tumors as well, since many of these seem to be caused by mutations similar to the one that causes CML.
What really is important about Gleevec is it's a paradigm, a cliche, overused word, but it really fits here, because what Gleevec represented was the first truly targeted therapy in cancer. In other words, where we took our understanding of the genetic abnormality that underpinned the cancer, we designed a specific therapy that would target that genetic abnormality, and that was present only in the cancer cells and not in the normal cells, and show that it could work.
We and others are interested in trying to extend these observations into breast cancer, prostate cancer. Investigators are looking at colon cancer for exactly these same types of mutations that cause the cancer, so that we can then design drugs that will turn those genes back off.
If we can do it in CML, hopefully we can do it in other cancers as well.
I'm really happy that there's more research going into that. And the fact that I feel so good and I'm able to do my normal things every day gives me a lot of hope about the future.
Thankfully, cancer is rare in children. In their short lives, they haven't had much exposure to things in the environment, like tobacco, that can cause cancer. And many adult cancers happen as a result of a number of changes to the cell, changes that take years to develop. Childhood cancer differs from adult cancer in other ways, too.
Probably the most striking difference or contrast between childhood cancer and adult cancer is childhood cancer is much more curable than adult cancer. If you look at all forms of childhood cancer, approximately 2 out of every 3 children who are diagnosed with cancer will not only survive their disease, but will survive in a healthy manner and live to grow up to be healthy adult. Whereas this survivability of adult cancer overall is much less than 50%.
Samantha Williams is a survivor.
Samantha is 10 years old, almost 11, very vivacious, fun. She's got a great sense of humor. I don't know if it's everything that she's gone through, but she's very patient, mature. Yeah.
Samantha's cancer, a brain tumor called a glioma, made itself known when she was at a medical lab getting tested for tonsillitis.
We're in the lab getting a strep test culture before school, and it was an immediate onset of a headache, just a severe. She was starting to pass out, didn't quite know where she was sweating, grabbing her head and screaming. And nobody was even helping, because that doesn't happen. And then she stretched out on the couch, and then it got kind of panicking. It was like, she's not okay.
Stacy rushed her little girl to the emergency room, where a CAT scan indicated bleeding in her head. She was flown to a larger, more specialized medical center.
And then they told us we need to take her to surgery right away and get a shunt in, get the pressure down. Her pressures were sky high in her head, and they said we have to take her and get that shunt in and, stop the bleeding. And I watched him come out, and he said, the bleeding's down, it hasn't bled anymore, and the pressure's down. And he said she was going to make it.
Samantha was then transferred to the University of California at San Francisco Medical Center. Here, doctors performed an exploratory surgery to find out what was really going on.
They did a biopsy, a full craniotomy biopsy, and it was wonderful news. I mean, to think you were happy for a tumor, but it was wonderful because they came out of the operating room, and it was like a scene in a movie. They came walking down the hallway. My husband and I are standing there, and the surgeons came walking down, pulled their mask down, and they gave me the thumbs up and said it was a benign tumor. And they knew exactly what it was. It was a juvenile pilocytic astrocytoma, and treatable. It doesn't kill children. It could eventually if you didn't treat it, because it follows the optic pathways and feeds itself around, but it was inoperable. They could not take it out. All right, Samantha, we have to make cookies here.
The tumor was wrapped around Samantha's optic nerve, her pituitary gland, and her internal carotid arteries. The risks of blindness or death were too high to risk removal.
So they said they couldn't take it out. So that began our journey with all the medications and, for the last 2 years. It's been our life.
Hey, gorgeous. How are you?
Over these 2 years, Samantha has undergone a range of different treatments as doctors tried to determine what would be effective for her. Samantha underwent 2 clinical trials. Though she would eventually be treated with radiation, her journey began with chemotherapy. Samantha's doctor advised a new approach, using 2 different chemotherapy drugs to shrink her tumor.
Virtually all chemotherapeutic treatments, which are frontline treatments, which are the primary treatment for tumors, are now combinations of different agents. Oftentimes agents that have completely different mechanisms of action so that they really target different aspects of the tumor cell's malignant growth. The other advantage is that virtually no drugs are 100% efficacious in killing all of the tumor cells, and so having combinations of drugs that work to kill tumor cells by different mechanisms means that you increase the chance of getting a higher percentage of the cells killed.
Doctors explained to Stacy that there were 2 possible treatments involved in the study.
We don't know which is better. We don't know which works better. And if you sign up to do the clinical trial, we will pick it out for you, it's random, which arm you will go on. You can stop at any time and choose which one you want. But you would be helping other children in the long run if we find out which one works better.
Headaches?
The decision to enter a clinical trial is never taken lightly, especially when children are involved.
It's very scary for a family who have just been told that their child has leukemia or some other form of cancer, to then also hear that we like to do some research, and perhaps that the treatment that we're going to give their child is research. Many families immediately translate this into sort of a guinea pig phenomenon and are concerned that we are experimenting on their child. We spend hours with families to fully inform them about the true nature of these clinical trials, in fact that their children will not be guinea pigs, and in fact the treatment that they'll be receiving has been tested and is felt to be not only safe but also effective.
And I gave her 3 different oral chemotherapy drugs every day for, I think it was about a week. And then they placed a portacath in her chest for the injectable chemotherapy. And we go over every other week on this cycle for injection to San Francisco of the chemotherapy. And then MRIs every 3 months with that. And that's for a year. And so we were very excited. She did beautifully on it. She never got sick.
But this was not to be the final treatment.
Her tumor had been stable all year, didn't grow, didn't shrink. They were happy with that. And then we did the MRI in April of last year, and the doctor had to give us the news that her tumor had grown.
At this point, Samantha's doctors recommended a different clinical trial, a phase 1 study of a treatment intended to alter the cancer-causing mechanism within the tumor cells.
It's not a chemotherapy agent. It is from what I was understood, it is a protein blocker for a certain RAS protein that they know is in tumors. We've all got RAS protein, but they know with these tumors that grow, these proteins get growing too fast, and it makes this mass. So this is a protein blocker. And they thought it'd be great for Samantha to try because it does not affect blood counts. It didn't even have a name yet. It's not FDA approved. It was SCH-66336 was the name of the drug. So we talked to Samantha and my husband, and we agreed that this would be a good thing to start. They had to have a spot open up for her. So we were excited about it. Not only, that would be good for her, but what an exciting thing if this turned out to be the cure for her kind of tumor, and kids didn't have to go through chemotherapy. They could go right on this drug and take care of the tumor.
Although this experimental drug has worked for some children, it did not help Samantha.
We started in June, and we ended in December. They pulled her off of it because in December we did another MRI and the tumor had grown again. And because she, was losing weight, and the doctor said it's not working for her.
By this time, Samantha's tumor was causing some symptoms. She had had several seizures, and the tumor was affecting her eyesight.
We had another eye exam, and at that point, they noticed that her vision was getting even worse in her right eye. So they felt that the tumor was probably pressing on her optic nerve at this point.
Samantha is old enough to understand what was happening, but she was not discouraged. Instead, she saw each new treatment as a cause for hope.
I went on chemo for a year, and then at the end, chemo didn't work. My tumor grew like 5 millimeters or something like that. And so then I went on an experimental drug, SCH-66336, and that gave me nausea, and I lost a lot of weight, which isn't a good thing. And then my tumor grew and my ventricles enlarged, so we got off of that. And then I got an internal shunt, which starts right here and drains my fluid into my stomach. And then I went on radiation.
Radiation was Samantha's last option. Her doctors believed it would be effective in destroying her tumor, but they were concerned about what else it might destroy. The tumor was pressed up against Samantha's optic nerve, so there was some risk of partial blindness. It also encompassed the pituitary gland, which regulates the hormones that control growth and normal development. So once they decided to go ahead, the radiation oncology team took great pains to make sure that the radiation was carefully aimed to reach just the tumor and nothing else.
Samantha's a quilt lady.
Ready?
She's not a big one quilt. Really?
All right.
Never brought that one.
Dr. Daphne Haas-Kogan is Samantha's radiation oncologist.
We used very sophisticated technology that we can offer here at our institution to target only the tumor and spare her other normal structures so that we cause very little harm to Samantha, but can still shrink her tumor and try to cure her.
They found out the exact location with the degrees of latitude and longitude that Dr. Hoskogan and the team had come up with. And then they formed the mask for her face. And then they tattooed the mask where exactly they wanted the beams to go.
Samantha got lucky in that she has a kind of glioma that behaves rather well. She has a low grade, a grade 1 glioma. So her chances of cure are actually very high. And we're very optimistic about her prognosis.
After 28 radiation treatments, Samantha's tumor is responding. The vision in her right eye is showing signs of improvement. The radiation can affect her pituitary gland, so she'll need to be watched closely by an endocrinologist.
And then we'll go every 6 months or so. Might go even more often once she hits puberty to watch those hormones. They all are replaceable. They can replace them either with shots or pills. We've gotten through it and we've learned a lot and we've all gotten stronger from it. And I hope that, we can reach out and help other people to show them that you do get through it. A diagnosis of cancer or brain tumor doesn't mean it's the end. And it can be okay. How'd it go? Good. She's an amazingly strong little girl. I couldn't be more proud of her.
All done.
Oh, we're stretch.
You tell Kelsey's family I said hi.
This story doesn't end with Samantha. In addition to working with patients, Dr. Hoskogan is also a research scientist working on new ways of using radiation in the treatment of brain tumors in children.
Okay? You're welcome. Thanks for everything, Patty. All right. 1, 2, 3.
For years, radiation oncologists have been improving the ways that radiation beams are aimed and focused, so the tumor is hit without damaging healthy tissue.
We are very, very good now at using computers and very sophisticated technology to target the tumor while sparing normal structures. We're now trying to make advances in the biological targeting of radiation.
So the same exploration of the molecular basis of cancer that led to the development of Gleevec is also opening doors for radiation treatment as well. Dr. Hoskogan and her colleagues are studying brainstem gliomas, which, unlike Samantha's, are usually fatal. One characteristic of these tumors is their resistance to radiation. They're very tough against any radiation bombardment. Dr. Hoskogan and her team are conducting a trial to see if a particular chemical can weaken the tumor cell's resistance to radiation so the radiation can kill it.
What we're testing in the trial is a novel agent in combination with radiation in the treatment of children with brain tumors. We're particularly enthusiastic about this agent because we think that it not only has potential on its own to perhaps shrink brain tumors, but also sensitize the tumor to radiation, which we already know is an effective agent against brain tumors.
Combining radiation with another form of cancer treatment like chemotherapy may seem like attacking the cancer on two fronts, hitting it with two big hammers, but it's not so brutal. The treatments actually complement each other. It's as if the chemotherapy strips the tumor cell of its armor, so the radiation can kill it with its sword. Dr. Norman Coleman is director of the Radiation Oncology Sciences Program at the National Cancer Institute.
Radiation and chemotherapy are highly interactive. If you look at cell killing curves of radiation alone, you get one curve. Drug alone may have very little cell killing. When you put them together, you get enormous enhancement in cell killing. So the chemotherapy is doing something that makes the radiation much more effective.
Beyond treatment, the other arm of progress in radiation research is in imaging, our ability to see into the body.
There are spectacular advances in imaging. In the past, most imaging has been shades of gray. One can try to image organs. There's a whole new field now called molecular or functional imaging, where one is actually trying to image the biological processes.
The leading tool in this advanced imaging is the PET CAT scanner. This woman has recurrent breast cancer. Doctors want to learn as much as they can about the tumor and see if it's spreading to other parts of her body so they can design a treatment. Before going into the scanner, she's had an injection of glucose, a type of sugar, that's combined with a radioactive agent that will be visible in the PET scan.
Cancer, for the most part, uses glucose as its source of energy to an extent that's much greater than surrounding tissues. So we can use PET as a way of detecting the presence of cancer based on the uptake of glucose or sugar by these tissues.
The PET CT scanner combines the image of tumor activity produced by the PET with a CT image.
The CT scanner is a way of creating a picture of anatomy. It shows you the structures in the body. It will not tell you directly how aggressive a tumor is or how much glucose that tumor is using. The PET scan shows you the level of glucose uptake, the level of intensity of its malignancy, but it doesn't show you the anatomy very well. So we can take the advantages of both modalities and combine them together by showing you the glucose uptake, tumor aggressivity, and the structure. How big is it? Exactly where is it? By superimposing it onto the CT image. The way this is done is we have a CT scanner and a PET scanner back to back within one housing. So we take the functional information that we get from a PET scan, superimpose it on the high quality anatomic information that we get from a CT scan to create one combined study.
Based on her PET CT scan, radiologists were able to see the spread of this patient's cancer, where a CT scan alone would not have detected it.
Okay, so we're looking at a recurrent breast cancer tumor, and what the PET scan is showing is the tumor metabolic activity, which in this case is very intense, and it's showing the extent of the disease into the chest wall. This small black spot on the scan indicates a lymph node that's involved with tumor.
The cancerous lymph node would have looked normal on a CT scan, but because the cancer was absorbing so much of the radiated glucose, it showed up on the PET scan. Superimposing the two images shows both the cancer and its location.
We're not just going to say, look, there's a tumor present. We'll be able to go to a subcellular level and tell you what's going on with this tumor. We're going to tell you what proteins are present. We're going to take advantage of certain components within a tumor and be able to image those. We'll know that by the components of this tumor, how are we going to treat it? The ability to diagnose different tumors and then to determine what the appropriate therapy is and how well that therapy is working will be something that will fall into the realm of molecular imaging. The possibilities are essentially limitless.
There are other promising approaches to cancer treatment that are also in the early stages of development. One of these is immunotherapy, using a vaccine to jumpstart the body's immune system so that it will seek and destroy cancer cells on its own.
Yeah, so I've got your cells here, and they looked really good in the lab, okay? The viability was really good, everything looked good with these cells, okay? So.
Unlike vaccinations for polio or the flu, which prevent you from getting ill in the first place, cancer vaccines are designed to kill cancer cells in people who already have the disease. Someday, cancer vaccines may be developed to ward off cancer entirely. This man has advanced colon cancer that had spread to his liver and was surgically removed. Even with the surgery, he only has a 1 in 4 chance of long-term survival. Surgeon Rick Barth is trying to improve those odds with a specially designed vaccine injected into the lymph nodes in the groin. This vaccine uses deactivated cells from the patient's own tumor to trigger the immune system into destroying any tiny developing tumors before they start to grow and spread.
As a surgeon, I take out most of their bulky tumor, and then I use those tumor cells to be able to form the vaccine. So instead of just having those tumor cells go in a bucket in pathology, we're using those tumor cells to be able to form this vaccine.
Yep.
Here we are.
Oh, beautiful.
We're also treating patients that have these little tiny tumors that, if left alone, will grow up and cause the death of the patient. That therapy is to try to enhance the patient's immune system so that the immune cells in the body can go anywhere in the body and potentially kill these little tiny tumor cells that may be somewhere else in the body.
Ordinarily, the immune system launches only a very weak attack against these cancerous growths because the immune system doesn't recognize them as foreign.
Unlike measles, and unlike the common cold, and unlike other infectious diseases, the malignant cell is us. And the immune system has always been taught during its development, don't make immune responses to yourself. But that means making the constructs transfecting and look at signaling.
Yeah.
Scientist Randy Noel has spent years building our understanding of the immune system. His lab has been studying ways to help regulate the immune system in specific ways to better fight disease. He's focused on dendritic cells, white blood cells that act as a kind of early warning device to activate the immune system against any foreign substance that could prove harmful, such as a virus or bacteria. These foreign substances are called antigens. When a foreign antigen is introduced into the body, the dendritic cells appear to gobble up the antigen, break it into pieces, then present the antigen on their surface. Ordinarily, this presentation or surface display by the dendritic cells tells other white blood cells called T cells to multiply and destroy the foreign antigen. But when the antigen is a cancer cell, this does not always happen.
We're trying to induce that patient's immune system to recognize the cancer cell as something dangerous and foreign, and therefore be able to react to it.
All right. Okay, so
Many scientists think that the dendritic cells may hold the key and are testing vaccines made from the patient's own dendritic cells in several types of cancer, including prostate cancer, kidney cancer, and melanoma. Dr. Barth wondered if this approach might also benefit patients with colon cancer. One day, he decided to attend a lecture by his colleague, Dr. Noel. Doctors Barth and Noel both work at the Norris Cotton Cancer Center at Dartmouth-Hitchcock Medical Center. Like all comprehensive cancer centers, it's a place that encourages doctors and scientists to interact.
I try to go to as many interesting lectures as I can. You know, it's tough because we're operating and, you know, doing experiments and, you know, taking care of patients, you know, most of the week. And, you know, so there'll be some, you know, lectures that will come up, though, that seem particularly interesting. And I was just fortunate enough to go to this one.
Dr. Barth listened as Dr. Noel described his work with autoimmune diseases like multiple sclerosis and rheumatoid arthritis, where the immune system goes into overdrive and attacks the body's own cells. Dr. Noel told how one of his graduate students had discovered a way to turn off the immune system by blocking the interaction between a molecule on the surface of dendritic cells called CD40 and a molecule on the surface of T cells called CD154 or CD40 ligand.
It's a needle in a haystack type of expedition. Sometimes you get lucky, and we got lucky. There was a communication link that really controlled the entirety of your immune response.
I think I talked to him right after the meeting, like right when he was done. I went up and talked to him about it. Randy's lab had an antibody that blocked that interaction between CD40 ligand on the T cells and CD40 on the dendritic cells. And he was working with models of autoimmunity, that is, where the times in your body where your immune system starts attacking your own normal cells in your body, and could show in those models that
use of this antibody blocked these autoimmune reactions. And while I was sitting there talking to him, I said, this has got to be important for cancer too.
The theory was that if blocking communication between CD40 and the CD40 ligand could block the total immune response, then increasing the interaction between these two molecules might stimulate the immune system into attacking the cancer cells that it usually left undisturbed. For the next 4 years, Drs. Barth and Noel tested this theory in mice. Through a series of experiments, they found that activation of CD40 on dendritic cells was required for many different anti-tumor vaccines to be effective.
So at this point, we were now really ready to go to patients.
But first, they had to submit their proposal to the Institutional Review Board at their medical center, a group that must approve all trials involving human subjects.
This is a group of scientists and clinicians that make judgments as to whether or not you should move ahead. And you present to them a wealth of scientific information, basic and maybe some preclinical data. And based upon that information, they say, go ahead or don't go ahead.
This Institutional Review Board, or IRB, is chaired by pediatric oncologist Eric Larson.
The IRB is dedicated to making sure that all clinical trials at that particular institution are safe and that the risk-benefit ratio for individuals who are interested in possibly participating is favorable, and that we are not putting patients at excessive risk without benefit.
Drs. Barth and Noel wanted to test a vaccine that mixed dendritic cells taken from the patient's blood supply with deactivated cells from the patient's tumor. They believed this mixture would intensify the dendritic cell's surface display of the tumor's identifying characteristics. Then, using a special technique, they would inject the vaccine directly into 2 lymph nodes in the groin. These lymph nodes contain large colonies of white blood cells. The goal is to activate the millions of T cells in these colonies to attack the tumor cells. Half the patients, chosen at random and without the doctors knowing which, would receive vaccine enhanced with a CD40 ligand to see if this added ingredient would heighten the immune response in humans the way it had in mice. The IRB gave the doctors permission to conduct a phase 1 and 2 clinical trial. They could now test the safety and effectiveness of their vaccine in 26 patients with advanced colon or rectal cancer. Claudette James was patient number 9.
What they did is they took the cancer cells from my liver and created a vaccine, and then they re-injected me in my lymph nodes with this vaccine. And being a very basic person, I always tell everyone, it's like if you get a snake bite, what do? They take the snake venom, they create the vaccine, and there you go, you know? So this, to me, in my mind, was basically the same type of thing, you know? And every cancer is individual, so if you're fighting it with your own individual cells, to me, it just makes sense.
These other tests were looking at your blood to see if there were some white blood cells in there that were reactive against your tumor. We compared blood samples before and after the vaccine, and the ones after the vaccine showed that we had induced an immune response in your blood against your tumor cells. So I just wanted to share that with you, okay?
Great,
But
that's
the study isn't designed to see whether this vaccine will improve the survival of patients with metastatic colon or rectal cancer. What the study is designed to do is to see whether we can induce immune responses in patients.
Claudette received 3 injections of the vaccine over a period of 2 months. Now she only returns to the medical center for follow-up exams. The trial has been completed and the results are being reviewed to see if the people given the vaccine with the CD40 ligand showed a stronger anti-tumor response than those who received the regular vaccine.
In terms of your eating and everything, is that okay?
I still can't, have a hard time because
The impact of either vaccine on long-term survival, if any, will not be known for several years.
we won't know results until 5 years because that's how long it takes, before they really get all the information. And at that point, they're supposed to let us know exactly what the statistics show. They don't give us good odds when we go into this because they can't, because it's not a proven thing. We were told that it was a 1 in 4 chance of a cure. But 1 in 4 chance sounds good when the alternative is 0. So that's why I felt like it was a well worth way of going. Because like I say, I think it's just another lifeline that you're given. It's a chance.
Every patient that Dr. Barth invited into the trial was willing to take that chance. But this was unusual. Although there are well over 1,000 clinical trials being conducted at any given time by the government, drug and biotechnology companies, and others, the participation of adult cancer patients in clinical trials is shockingly low.
Unfortunately, with regard to adult cancer, only about 3 to 5% of cancer patients participate in clinical trials. There are a lot of reasons why that is the case. There's no one simple answer. Therefore, there's not one simple thing that will change, that'll radically, significantly increase that number. But we must increase that number. We need to have more adult patients participating in clinical trials because we need to be able to rapidly accelerate our ability to improve and develop more effective interventions.
This is the liver here, okay? And back here is where I did the surgery, and those white things are just clips.
Dr. Barth had already successfully removed his patients' liver tumors, which perhaps made them more willing to enter his experimental trial. But there were other reasons, too.
I could tell patients that patients were not having, that were being treated, were not having much in the way of toxicity, and so most patients looked at it as basically a bonus. It's a little inconvenient to come to the clinic to see me 6 or 7 times to get the cells grown, to get the injections, to be checked by me. But that really, other than the inconvenience, there wasn't much of a downside to it, and there was, potentially substantial benefit to be gained from it.
Take a look at your hands, Mr. Willis. Okay.
The outcome of any trial is unknown at the start, and there's always the possibility that the new treatment will not work. But patients are closely monitored throughout to check for any possible negative effects, and this can be an advantage.
Some patients have trepidation about clinical trials. They're afraid they're going to be, quote, guinea pigs, unquote, something like that. But in point of fact, people get extremely good care if they're on a clinical trial because they're monitored very closely, usually in the care of doctors who've been doing, or experts in the field.
Participating in a clinical trial offers another benefit as well: the chance to have a personal impact on the direction of medical progress.
Clinical trials are an essential part of establishing the efficacy of the effectiveness of any new treatment.
One cannot give guarantees or assurances that for a particular patient, that new intervention is going to be of benefit to them, although the data and the experience may ultimately be used to benefit everyone.
It's inspiring to me personally, the bravery actually of patients in facing a difficult situation and still choosing to try and take part in a process that will certainly yield new knowledge for the world at large, as well as hopefully benefit them.
The most interesting and important question for me about a tumor is, number one, why did it arise in the first place? On a molecular level, on a genetic level, what are the aberrations, what are the mistakes on the genetic level that have led to this tumor? And number two, in order to benefit my patient, I need to learn how to target that mistake, that accident, that genetic aberration that occurred, so that I can turn back the clock and make this tumor now, make these cells now behave in a normal fashion, so that they no longer threaten my patient's life.
cancer treatment should be based upon the discovery of knowledge and the use of that knowledge to make new discoveries. And that's what's going on bit by bit. Now, there have been a tremendous acceleration of progress in our understanding of cancer. And I don't know where this is going to lead, except that the more we know, the better we do.
In the old days, science was sort of fishing in the banks and fishing in the trees. We're actually fishing in the water now for the answers. Where we're looking for the answers is really where the answers lie in basic fundamental biology. So it's just a very exciting era. Combined modality therapies will look completely different in the next 10 years than they did in the